Hello everyone. In this video, we are going to talk about the COSMID vectors. So, we will look into this video what are the COSMID vectors. So, basically these COSMID vectors, they are actually the plasmids that carry cos sites. So, we can say that these COSMIDs, they are the vectors that are hybrids of lambda phages and plasmids, and their DNA can replicate in the cell like that of a plasmid, or can be packaged like that of a phage. So the advantage here is that these COSMID vectors, they have the high transformation efficiency, and they carry up to 45 kb of the DNA insert. So we will see here this COSMID vector. It contains the multiple cloning site, it contains the cos site, it contains the resistance gene, that is the ampicillin resistance gene, and it has the origin of replication, that is the call even origin of replication. So these cosmids, they are the cos sites along with the plasmid DNA. They carry actually the origin of replication, the ampicillin resistance gene, then the cos sites, then the polylinker site. So we can say that the development of COSMID vectors, this was actually based on the observation that about 200 base pair DNA sequence in the lambda genome, that is called as the cos site, that is actually required for lambda DNA packaging into phage particles during the lytic infection. So these cosmids, they are the modified version of plasmid, and they normally carry 45 kb of DNA insert. And these cosmids, the cleavage at cos site by the lambda terminase protein during the phage packaging, it produces actually the 12 nucleotide cohesive end at the termini of the linear lambda genome. So recircularization of the lambda genome after bacterial infection, that is actually facilitated by base pairing between the complementary cohesive ends. So here we have these recombinants, they get packaged in the head of the bacteriophage, while those that get self-ligated, they don't get packed. So we can say that for in vitro packaging of DNA into lambda phage, the only requirement is the presence of two cos ends that are actually separated by 37 to 51 kb of intervening sequence. Most cosmids are about 5 kb in size, and cosmid cloning vectors with DNA inserts of 30 to 45 kb can be packaged in vitro into lambda phage particles. So the transformation efficiency is very high. So phage is very efficient in terms of infecting bacteria. So as compared to other types of the transformation methods, this method is very efficient as compared to other methods, as it efficiently infects bacteria. So if we can see here the cleavage at polylinker site, we can use the target DNA, then the linear cosmid, so that we can make the recombinant part of the cosmid, and that construct is then packaged in phage head so that it can infect the E. coli cell. So we can insert our gene of interest with the help of the phage infection. So this is the actually the cosmid vector. So E. coli cells, they infected with the cosmid packaged phage, they are able to support autonomous replication of circular plasmid. So this is the cosmid vector.